Biology Exam Preparation for Medicine Students Part 4. Which type of cell junction allows ions to pass directly between adjacent animal cells?
The correct answer is gap junctions. Gap junctions are specialized intercellular connections that directly link the cytoplasm of two adjacent cells, allowing ions, nutrients, and other small molecules to pass freely between them. These junctions are formed by proteins called connexins, which assemble into a channel structure known as a connexon. This setup is particularly important in tissues requiring rapid communication, like cardiac muscle and certain types of smooth muscle, where electrical signals need to propagate quickly for coordinated contraction. In contrast, tight junctions create a seal that prevents the passage of materials between cells. Desmosomes provide mechanical strength by anchoring cells together. And adherence junctions connect the actin cytoskeletons of neighboring cells, but do not facilitate direct communication like gap junctions.
Which of the following processes occurs in the stroma of chloroplasts?
The correct answer is the Calvin cycle. The Calvin cycle is a series of enzyme-driven reactions that occur in the stroma, the fluid-filled space surrounding the thylakoids in chloroplasts. During this cycle, carbon dioxide is fixed and converted into glucose using the energy-rich molecules ATP and NADPH, which are produced during the light reactions of photosynthesis. This process is also known as the dark reactions or light-independent reactions because it does not directly require light. In contrast, the light reactions take place in the thylakoid membranes. Photorespiration is a wasteful process that competes with the Calvin cycle, and photolysis refers to the splitting of water molecules during the light reactions.
Which DNA repair mechanism is specifically used to repair thymine dimers caused by UV light?
The correct answer is nucleotide excision repair. Thymine dimers, caused by UV light exposure, result in covalent bonding between adjacent thymine bases on a DNA strand. This distorts the DNA structure and impedes replication and transcription. Nucleotide excision repair is the primary mechanism that corrects this damage. It involves recognizing the distortion, excising a short segment of the damaged strand, and then using the undamaged complementary strand as a template to synthesize the correct DNA sequence. Mismatch repair fixes errors that occur during DNA replication. Base excision repair removes single damaged bases, and homologous recombination is primarily involved in repairing double-strand breaks, not thymine dimers.
What is the primary role of histone proteins in eukaryotic cells?
The correct answer is to organize DNA into chromatin. Histone proteins are essential for compacting and organizing DNA within the nucleus. DNA wraps around histones to form nucleosomes, which are the basic structural units of chromatin. This organization not only condenses DNA to fit within the nucleus, but also regulates gene expression by controlling the accessibility of DNA to transcription machinery. Histones do not catalyze DNA replication or repair DNA mutations. While they influence RNA transcription indirectly by altering chromatin structure, their primary role is structural organization.
Which of the following is a hallmark of facilitated diffusion?
The correct answer is that it requires carrier proteins. Facilitated diffusion is a passive transport mechanism where specific carrier or channel proteins help move molecules across the cell membrane down their concentration gradient. This process does not require ADP because it relies on the natural kinetic energy of molecules. In contrast, active transport requires ATP and moves solutes against their concentration gradient. Facilitated diffusion is a universal process and is not limited to prokaryotes.
In fungi, what is the term for the thread-like filaments that make up the mycelium?
The correct answer is hyphae. Hyphae are the long, thread-like structures that collectively form the mycelium, which is the vegetative growth of fungi. Hyphae grow and branch extensively, allowing fungi to absorb nutrients from their environment. They play a crucial role in decomposition and nutrient cycling in ecosystems. Spores are reproductive structures, rhizoids anchor certain fungi to their substrate, and conidia are asexual spores produced by some fungi.
What is the primary function of the wobble hypothesis in translation?
The correct answer is that it promotes redundancy in the genetic code. The wobble hypothesis explains how a single tRNA molecule can recognize multiple codons for the same amino acid. This is because the third nucleotide position in a codon often exhibits flexibility, or wobble, in its pairing with the anticodon of tRNA. This redundancy enhances the efficiency of protein synthesis and reduces the likelihood of errors. The wobble hypothesis does not regulate tRNA synthesis, ensure transcription fidelity, or increase RNA stability directly.
What type of molecule is the primary component of bacterial cell walls?
The correct answer is peptidoglycan. Peptidoglycan is a polymer composed of sugars and amino acids that forms a rigid, mesh-like layer around the bacterial cell membrane. It provides structural support and protection, helping bacteria maintain their shape and resist osmotic pressure. Chitin is found in fungal cell walls, glycogen is a storage molecule, and cellulose is the main component of plant cell walls.
Which organelle contains enzymes that break down hydrogen peroxide?
The correct answer is peroxisome. Peroxisomes are specialized organelles that contain enzymes like catalase, which break down hydrogen peroxide into water and oxygen. This process protects the cell from oxidative damage caused by reactive oxygen species. Lysosomes are involved in digesting cellular waste. The Golgi apparatus modifies and sorts proteins, and endosomes are involved in the transport of materials within the cell.
In genetics, what is the term for the phenomenon where one gene affects multiple traits?
The correct answer is pleiotropy. Pleiotropy occurs when a single gene influences multiple, seemingly unrelated traits. For example, mutations in the Marfan syndrome gene can lead to changes in the cardiovascular system, skeletal system, and eyes. This is distinct from codominance, where both alleles in a gene pair are fully expressed, epistasis, where one gene affects the expression of another, and polygyny, where multiple genes influence a single trait.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.